Okay. Hello, everybody. Today we're going to be talking about genetic variation, which is a topic that we've discussed somewhat a lot already, but we're going to go into some detail here about the causes or factors that result in genetic variation. You can see them listed here. We're just going to briefly talk about them here in this presentation, then we'll talk about them in more detail as the unit unfolds. All right, but you can see here mutations, crossing over, independent assortment, and sexual reproduction are the methods we're going to be discussing here. So, as you can see, and as we've talked about a lot, right, variation is important because without these changes, right, these differences, um, a species can go extinct all by the same thing, right? So it's important to have differences so that you build up um, a bit of variety so that creatures and members of your species and population will survive no matter what. So keep in mind, as we talked about in the last unit, that genes have usually at least two forms or what we call alleles, right? So because of this, we have what you might call invisible variation because a difference in allele is a genetic variant, right? Like that is a variation. So it doesn't have to be a physical trait or the phenotype where this is expressed. It can simply be a genotype difference. And so if you are heterozygous, um, it may be like it says here, invisible. So keep in mind that's one of the reasons why this is a lot more challenging to conceptualize because it's a lot of times hidden. Um, Keep in mind, as we've brought up before, this is relative to a gene pool. So if you think about how we talk about evolution, um, we talk a lot about how it's not individuals, right? It's a species that will see these changes. Well, we typically call that just a population, which, as you can see here, is a group of individuals of the same species that interbreed, meaning that they're in a close enough proximity, right? They're in the same area. Now, if you consider all of those members of the population, right? If you take all of their genes, right, all of their alleles, that would be called the gene pool, and that is where those differences would add up, right? That we were talking about in the last slide. So, when we talk about the relative frequency of an allele, this is where we would have a percentage, right? Where we would be able to calculate some sort of accurate or semi-accurate count of the number of alleles of each type that occur in a gene pool, right? So you compare them to the other ones. And since this is expressed as a percentage, it's not related to dominant or recessive. Okay, we talked a lot about, for example, polydactyly and having more than five fingers or on your hands or toes, right? The reason why that is the dominant trait, but not the most commonly seen, is because it is not common in the gene pool, right? So this idea of relative frequency is really important because this is how we define evolution. Okay, so when we think, as it says down here at the bottom, we determine whether a population is evolving. Oftentimes, they look at the sum of the population's alleles or its gene pool. Okay, so if you look down here in this diagram, you see that 48% of these rats were black, right? Whereas you have 16 were homozygous black. So all in all, right, 64% are going to show the black trait, even though there's a bit more of that brown trait available. So this is how we would represent a population's gene pool, right here, and we would track the relative frequency of these alleles, right? The brown fur and the black fur allele. Okay, and as you can see, it's a bit more, it's a lot more even than it might look from the 64-36 difference in the coat color. Okay, so keep in mind, gene pool matters, and this is where a lot of these differences are experienced. Okay, evolution is simply a change in the relative frequency of alleles in a population, as we talked about. A significant enough change is what we will define as a population evolving. Okay, so simpler than maybe some make it out to be. So again, that's the end of the story, but we need to continue to talk about the beginning. So, what are the sources of genetic variation? These pathways that create the differences of the species, of the members of the species, so that they can survive longer. Well, one very common way that students are familiar with, even though we haven't talked yet about the specifics here, you should know or probably have heard about a mutation, a DNA mutation, right? So, anytime we have a change in the sequence of DNA, right, we're going to cause a change in the gene, potentially, if that's where the mutation occurs. And then a mutation of a gene is going to mutate the trait that shows up with it, right? So minor changes in DNA can lead to minor changes in genes that are significant in an organism's appearance or function or physiology or something like that. So anyway, hopefully it makes sense that a tiny change in genetic sequence can add up to a pretty significant physical difference in the trait, right? So these mutations occur because of mistakes in DNA replication or as a result of mutagens. These are things that you introduce yourself to during your lifetime, right? Radiation, chemicals, smoking is a cause of this kind of mutagen source, right? For example. So keep in mind, they don't always affect phenotypes. Just like before, we were talking about these invisible variations. Here, mutations sometimes are quote unquote invisible, right? Because they are present but not affecting a gene or not changing the trait. We also have this concept of gene shuffling. Now, this is a bit trickier because we haven't talked a lot about meiosis, but you did talk about it in middle school, right? So how do genes get shuffled? Well, there's a couple different ways. Sexual reproduction is a short way of talking about this, right? Because the process of sexual reproduction, as opposed to like asexual reproduction, is where we see the rise of these gene shuffling methods, right? So the first idea that again, I think you can appreciate is the fact that there are a tremendous amount of sperm that are released when fertilization is taking place, right? And so which sperm actually gets to fertilize the egg is random, right? None of them are pre-programmed to win, if you will, this race. So there's some randomness there, right? So the random fertilization of gametes is what we call that. Okay. We also have what's called the law of independent assortment. Okay. And as it says here, the law of independent assortment is when alleles in meiosis segregate independently of other pairs of alleles during the gamete formation. So in other words, right, you've got a lot of traits. And as we've talked about, you only give half to an offspring. So when you're forming your gametes, sperm or an egg, depending on male or female anatomy, right? When you are creating those, you're only going to give half your traits. And each trait is not, for the most part, right? They are, well, not for they are completely separate of each other, right? So which trait goes with the other, again, which hair color versus which eye color versus which, length of your big toe trait, right? These are all things that are gonna segregate, right? So as it turns out, the ways, the combinations of the genes that your parents have when they form you, right, are even more complicated than just the fact that you only get half and half, right? These things can sort in very, very unique ways, and so this is an important aspect of what we call gene shuffling. Okay. Oh, here's a picture of the gene shuffling process. So if you just track here are the alleles in the different chromosomes, right? So you see here we would have in meiosis, which is the process of making a sperm or an egg cell. Okay, here we would have the original, and then here we have the split, right? So we have metaphase one, where one of these two things would occur, right? It's random which one of these occurs. Okay, but take a look at the different gametes that would form based on just which one of these random events took place. So you can see here that the combinations are completely different from one simple difference. And so again, independent assortment allows for a lot of different variation of the genetics, right? The genome, which again is important for genetic variation. This is why we do the foiling of the gametes in dihybrid crosses, right? So this is why Punnett square is valuable here, because we're actually demonstrating the law of independent assortment by simulating all these different gametes and how they might look. So we are figuring out what are all the different ways that they could independently assort. That's why we foil. So another method that gene shuffling can take place in, and one that you've again may have mentioned in middle school, is this process called crossing over. So when your chromosomes are forming, right, during this process of meiosis, what happens is sometimes they break off, and when they're forming, they create these new combinations of genes, right? So, again, here's an example of what. Move this out of the way. So down here in the bottom left, okay, you can see it says 2 homologous chromosomes pair up with each other during prophase 1 in meiosis. So again, a homologous chromosome are chromosomes that are identical in what they contain, right? They can be different versions, but they're going to contain the same genes. So as it says here, right, you can see sometimes these things become entangled, and then they break off, and they actually crisscross what they have on them, the information they carry. So this information would come from different parents. Therefore, we would have whole different combinations of chromosomes that could be created. And so this is a very significant, and this happens, every time this process takes place, right? Every time during meiosis. So anyway, here is a visual example again of what we call genetic recombination through crossing over. Okay, so again, this information is the same thing as I just said on the last slide. It's just putting it in a bit better visuals. Okay, so let us know if you have any questions, right? Let me know. This topic is one that, like I said, we'll keep discussing, but for now, that's all I got. All right, you guys have a good rest of your day.